Hopefully, although this is scientific, you will still retain a bit of energy to see you through until lunchtime at 1 o'clock. I don't know about you, but I do wilt without food. So hopefully your caffeine and your biscuits will see you through. As you can see, my name is Elaine Vickers, and I'll first of all just give you a quick introduction to who I am, and then begin my talk. My background is as a molecular biologist. I did a PhD up at the University of Manchester, and then went straight to work for Cancer Research UK in their science information team, helping them make their research and their science understandable to the outside world. I spent many years there, but for the past 6 years, I've been self-employed, and I spend my time often alone, trying to get to grips with all the science, all the research that's taking place, and turn that into understandable, hopefully informative, hopefully useful and memorable presentations that demystify, that unravel, that explain the science behind the brand new cancer treatments that are being developed and coming through trials all the time. Today I've been asked to speak about cancer immunotherapy. This, over the past 5 or 6 years, has suddenly taken the scientific world and then the medical world and then just kind of the world in general by storm. It's a very different way of thinking about cancer than we're used to in talking about chemotherapy or even some of the newer targeted therapies. It requires a different spectrum of knowledge and understanding to get to grips with than some of the other treatments that you might be more familiar with. So this morning, or this afternoon, whichever way you want to look at it, I'm giving you 3 main topic areas. So a bit about cancer cells and the immune system, how cancer cells dwell alongside and interact with and suppress the immune system. Then I'll talk about what is cancer immunotherapy in that case. What is it there to do? What's the goal of it? And I'm then going to focus on 2 areas of cancer immunotherapy. I'm going to talk about antibodies as cancer treatments, and then CAR-T cell therapy, often something that ends up in the newspapers, not always something that's explained particularly well. So I'm going to talk about those 3 topic areas. We'll start off with a few basics about cancer cells and how they interact with their immune system. Cancer cells are essentially cells in which their instruction manual has gone wrong. They no longer behave normally because their instructions telling them how to behave normally have become corrupted. The instruction manual of a cell is its DNA, this long spiraling molecule that sits inside the nucleus, the heart of every cell. I always find it incredible that although cells are individually too small to be seen by the naked eye, they each also have 2 meters of DNA inside them. It's a very fine strand. It is packaged incredibly tightly and incredibly well to form these structures that we call chromosomes. There should be 23 pairs of chromosomes in every cell in our body, apart from the red blood cells, which just carry oxygen and don't have any chromosomes. So all the other cells in our body contain this set of instructions in the form of DNA. If that DNA becomes corrupted, then the instruction manual is wrong and the cell no longer performs its functions normally. And the instruction manual, that DNA, is there to tell the cell how to make proteins. And it's the proteins in our cells that do all the work. There are tens and thousands of different proteins in every cell. Each protein has its own role to play. They come in different shapes and sizes. Some of them are structural, some of them are mechanical, some of them are catalysts making chemical reactions happen. It's the enzymes that do the stuff in our cells. And if the DNA is corrupted, then the proteins are corrupted. And because the proteins do all the work, if the proteins are wrong, then the cell behaves wrongly. In general, the cancer cells of childhood cancers have fewer mutations, fewer faults in the DNA than the cancer cells of adult cancers. There is usually one fault, one mutation, which sets the wheels in motion. For example, in an acute leukemia, it's often the presence of a fusion protein. That sets the wheels in motion. Later mutations follow. Ultimately, the cell becomes a cancer cell, but it's still genetically less complicated than an adult cancer cell. But cancer cells all have one thing in common, and that is that they are unstable. They collect new mutations as time goes on, and they diversify as time goes on. So when someone is diagnosed with cancer, it's not that they have one population of cancer cells in their body, they may have many. And each population of cancer cells, because it's unstable, because it's accumulated new mutations as time has gone on, each cancer cell has its own unique combination of mutations inside it. When we talk about chemotherapy, we're generally talking about drugs that target cells that are trying to multiply. When we talk about radiotherapy, we're talking about beaming high energy rays at the cancer in the hopes of breaking apart its DNA. When we talk about targeted therapy, we're often talking about understanding what particular mutations have occurred in the cell's instruction manual and what drugs we might have that can target those particular mutations. But when we talk about immunotherapy, we're talking about drugs that disrupt or take advantage of the patient's immune system and the way the cancer cells are interacting with their immune system. It is possible to use the immune system against cancer because the immune system often does know that cancer is there. And in order to survive, in order to thrive, in order to multiply, the cancer cells have found a way to suppress, to evade, to hide from the immune system. And cancer cells, particularly those cells in tumors, they live alongside, very happily, some types of white blood cells. For example, macrophages. I think of them as the jack of all trades of our immune system. We have many, many different types of white blood cells that together make up our immune system. Each white blood cell attacks invading organisms like bacteria and viruses and nasty little worms. They attack them in all sorts of different ways. Each white blood cell has its own specialized role. But the macrophages are kind of the jack of all trades. They're there as a first line of defense to munch up and destroy invaders. They're there to support other white blood cell types and help them sustain an immune reaction. They are also there as a pivotal player in inflammation. In cancer, their presence is often a bad sign. It's often a sign that there is some kind of inflammatory reaction going on within the tumor that the tumor is harnessing for its own goals. So the inflammatory white blood cells, the inflammation-associated white blood cells like the macrophages, like mast cells, neutrophils, eosinophils, basophils, they're often doing us no good. They're sitting within the tumor, they've been co-opted by the cancer cells, they've been changed, they've become useful to the cancer cells, and they have become part of the problem. Also part of the problem are the myeloid-derived suppressor cells and the regulatory T cells. All these white blood cells have normal, healthy functions, but within a tumor they've been co-opted and persuaded by the cancer cells to take on abnormal roles. And the myeloid-derived suppressor cells, the regulatory T cells, they again aid and abet the cancer cells. Cancer cells also hide from the immune system. Normal, healthy cells put all sorts of little protein fragments on their surface. They display them there in order to be open and accountable to the immune system, to say to the immune system, this is what I'm doing. I'm showing you exactly what proteins I'm making by displaying fragments of them on my surface. And the white blood cells, they come along, blood cells like natural killer cells and cytotoxic T cells, they crawl along inside our tissues. They know, okay, this cell is healthy. It's showing me normal proteins, normal fragments. I can leave these cells alone. Cancer cells often lack the normal proteins from their surface, and they hide from the immune system. They edit what they put on their surface in order that they don't get seen by the natural killer cells and cytotoxic T cells. You also get a phenomenon called tolerance, where the immune system has seen the cancer cells for so long, over such a long duration of time, that they are essentially shrugging their shoulders and saying, so what? We're used to this. It looks normal to me. I don't care anymore. And that's called tolerance. Another thing that cancer cells do is that they suppress any good white blood cells that try to destroy them. Cytotoxic T cells are often the good guys, as are dendritic cells. These are ones that together can create a cancer-fighting immune response. but the cancer cells suppress them directly, and they use these myeloid-derived suppressor cells, the regulatory T cells, to suppress them on their behalf. And together, they completely incapacitate any regulatory T cells and dendritic cells that would otherwise be working together to mount an anti-cancer immune response. And the goal of immunotherapy, this new generation of cancer treatments, the goal of them is to overcome the suppression or create a cancer-fighting immune response from scratch, or to target the way the cancer cells are interacting with the white blood cells. They are there to target the immune system, not directly the cancer cells, and to use the immune system to fight back against that patient's cancer. There are three main types of cancer immunotherapy. One type of cancer immunotherapy is to use antibodies. To use antibodies either to provoke an immune reaction against cancer cells, to kill cancer cells directly, or to prevent cancer cells from suppressing white blood cells. I've highlighted kill cancer cells directly because it's up to you to decide whether that still constitutes immunotherapy. Antibodies, as I'll just explain in a second, are part of our immune system, and some people would say any antibody-based treatment is essentially an immunotherapy because antibodies are part of our immune system. Other people would say, no, actually, if we're using an antibody to kill cancer cells directly, we're no longer doing immunotherapy. It's up to the individual. Different scientists, different doctors have a different opinion on that. Then we have cell-based immunotherapy, using the patient's own white blood cells to fight their cancer. And this often involves taking them out of the body, modifying them in some way, and putting them back again. We also have vaccine-based immunotherapies. I'm not going to talk more about them today. They're still very experimental. We haven't got any licensed vaccine-based immunotherapies, although it depends on what you say is a definition of a vaccine. That varies. But essentially, the vaccine-based immunotherapies are to do with encouraging the patient's own white blood cells in situ, still in their body, to attack and destroy cancer cells. So you're not taking their white blood cells out and reprogramming them and putting them back. You're working with the white blood cells that exist and trying to find a way to trigger them into action. I'm going to talk about first, antibodies as cancer treatments. And I'm not going to presume that you automatically know what an antibody is. I'm sure you've heard of the word. I'm sure you've got some idea of what it is, but I often think it does no harm to refresh people's knowledge, especially in a context like today. I thought I would just refresh your knowledge of what an antibody is. To understand antibodies, we need to understand a little bit about our body's B cells. Our body's B cells are one type of white blood cell found in our body. B cells are made in the bone marrow, hence the letter B. When they're made in the bone marrow, they gradually mature in the bone marrow and they start putting what is called the B cell receptor on their surface. And each B cell has its own unique B cell receptor. It makes thousands of copies of it and it puts it on its surface. Once that B cell is mature and it's got its B cell receptor on its surface, it goes out to where it might come across an infection. So it goes into the spleen, it goes into lymph nodes throughout the body, it goes into the linings of the intestine. It hangs out where infections happen. And if that B cell comes across an infection that fits precisely the B cell receptor on its surface, it may then become an activated B cell, and it starts releasing its B cell receptor. It often moves to the bone marrow, goes back to the bone marrow in order to do this. It sets up shop back in the bone marrow, releases its B cell receptors into the bloodstream, but now we don't call them B cell receptors anymore, we call them antibodies or immunoglobulins. But they are B cell receptors, but now in the bloodstream. And in the bloodstream, the antibodies travel around and they support various aspects of our immune system. And one really critical property of antibodies is they lock on very tightly to their target. They only lock onto one target, they will never lock onto any other. And because of their specificity, scientists for many years have argued that antibodies would make great cancer treatments. If we could create antibodies that didn't latch on to bacteria and viruses, but instead latched onto proteins on the surface of cancer cells, wouldn't that be helpful? because those antibodies would bring in other white blood cells, would activate them, would create an immune response against the cancer cells. And that's what scientists have been doing now for decades. One way of doing that is to take proteins from the surface of a cancer cell, put them into a mouse, let the mouse's immune response get going, let the mouse produce antibodies against that cancer protein, use the mouse antibodies as a cancer treatment. That doesn't work too well because the mouse antibodies are visible to the patient's immune system. The patient's immune system recognizes the mouse antibodies as something weird and wacky and foreign and starts destroying them. So instead, scientists now create modified antibodies that are less detectable to the patient's immune system. And one quick note on the naming of things, we call them monoclonal antibodies because the antibody in an antibody treatment, all the antibody molecules in that treatment are identical to one another. They've been made by one population of genetically identical cells, one clone of cells, therefore they're called monoclonal antibodies, one clone antibodies. They're all identical to one another. The name of an antibody reflects somewhat what that antibody's been made from. If it's a mouse antibody protein, then there's an O in the name. If it's part mouse, part human antibody, it's an XI. If it's mostly human, a little bit mouse, there's a ZU in the name. If it's fully human, there's just a U. And antibodies, some of them can kill cancer cells outright. They can attach to a protein on the surface of cancer cells. If that protein is necessary for the cell's survival, then blocking it might kill that cell. It's up to you whether you call that antibody a form of immunotherapy or not. Others can't do that so well just as they are, but you can use antibodies as a delivery device. You can attach chemotherapy to the back end of an antibody. The antibody latches onto the cancer cell. It gets drawn into the cancer cell. The chemotherapy part of the antibody is released. It kills the cell. These are called the drug-conjugated antibodies. Brentuximab vedotin is a licensed treatment for Hodgkin lymphoma. It is one of these conjugated antibodies. But antibodies can also trigger an immune response. They can attract in macrophages, these jack-of-all-trades. Some macrophages, if they've been altered too much by the cancer, they won't be able to do this, but fresh macrophages drawn into the tumor, they might be able to do this. They might be able to kill the cancer cells by being drawn in by the antibodies. Natural killer cells as well, they can be drawn in by antibodies, activated by them, and then the natural killer cells and the macrophages directly kill the cancer cell to which they've become attached. Antibodies also attract what are known as complement proteins. The complement proteins are a set of 20 or so proteins found in our bloodstream which support and help the immune system. The degree to which an antibody can draw in and activate white blood cells is going to vary from patient to patient. In some tumors, the cancer cells are so good at suppressing white blood cells that try to attack them that this can't happen. But in other cancers, the white blood cells are a bit more awake, they're doing a bit more stuff, they can be attracted by antibodies, they can destroy the cancer cells. I also thought I'd mention one novel way of using antibodies as treatments, and that's to create a bispecific antibody. A treatment made of the two little end portions of two separate antibodies, fuse them back to back, and then use them to bring together different types of cell. I've given you here in the example of blinatumomab. This is back-to-back fusions of a part of an antibody that targets the CD3 protein found on T cells and another antibody that targets a protein called CD19 found on B cells, including cancer-causing B cells. And the bispecific antibody draws the two cell types together and activates the T cell that it now kills the B cell. I also wanted to describe to you CAR-T cell therapy, partly because it generates so many column inches. Everyone gets very excited about it, but it is quite complex science. CAR-T cell therapy rests on T cells. They're a bit like B cells, but they're a bit different. They're not made in the bone marrow, they're made in the thymus, hence the letter T. They don't have B cell receptors on their surface, they have T cell receptors on their surface. And when they fight infections and fight cancer cells, they don't do it by releasing the T cell receptor into the bloodstream, they do it by direct hand-to-hand combat. There are three main types of T cell: cytotoxic T cells, helper T cells, and regulatory T cells. And it's the cytotoxic T cells that we're interested in because these are the ones that have the potential to kill cancer cells. Often they don't do that. Often it's because actually the body doesn't contain any cytotoxic T cells that know what a cancer cell looks like and know how to attack it. So sometimes the body just doesn't create cancer-fighting T cells. Sometimes the body has created T cells that have the potential to fight cancer, but they haven't done so because the full activation process hasn't happened for that T cell. The full activation process requires dendritic cells, and for whatever reason, that hasn't taken place. Or even if cancer-fighting T cells have been made by the body, they've entered a tumor, but now they're being suppressed by those myeloid-derived suppressor cells, by those regulatory T cells. So the body's trying to fight back. It's made some active T cells, but the T cells can't do the job they were designed to do. So the purpose behind CAR T cell therapy is to overcome those three obstacles, to create T cells deliberately that can recognize a cancer cell, to create T cells that no longer need dendritic cells in order to become active, and to create T cells that are so active that they won't get suppressed by the myeloid-derived suppressor cells or the regulatory T cells. The basic idea behind CAR T cell therapy is that you take some T cells from the patient's blood, you modify them in the lab so that they now know how to fight cancer cells and no longer need dendritic cells to become active. You allow them to multiply, you put them back into the bloodstream, you hope that they will track down and destroy the patient's cancer cells. They are genetically modified so that they produce a modified T cell receptor that means the T cells no longer require dendritic cells. So the key thing with CAR T cells is that you modify them with this CAR protein, a chimeric antigen receptor. And that chimeric antigen receptor, there's bits taken from T cell receptor protein, there's a bit taken from an antibody, they've been fused together. And what you do is you take T cells from the patient's bloodstream. You create a weird virus particle where you've emptied out all the normal disease-causing virus bits and you've replaced them with the instructions for making that CAR protein. You infect the T cells with the virus. The instructions for making the CAR protein gets incorporated into the T cell's DNA. The T cells then manufacture lots of that CAR protein and they display it on their surface. You return the T cells to the body and they track down and destroy the cancer-causing cells. Normally, at the moment, the protein on the cancer cells that the CAR protein is designed to lock onto is a protein called CD19. And CD19 is found on the cancer cells of B-cell cancers, B-cell leukemias, B-cell lymphomas. They will have that CD19 protein on their surface that the CAR attaches to. They will, of course, kill healthy B cells as well as the cancer-causing B cells. They both have that CD19 protein on their surface. So far, trials have been fairly small, generally 15 to 50 patients, occasionally now up to 100 patients. Most of the trials have been in children and adults with B-cell leukemias or lymphomas, people for whom all other treatments have already failed. The remission rates are high, which is why people get really excited about them. Remission rates are generally 60 to 90%, but there is generally later relapse. The cancer hasn't gone away for good. It often does come back, but now the cancer cells lack that CD19 protein on their surface, so adding in more CAR T cells is not going to help because they can only kill cells with CD19 on their surface. And there are also dangerous side effects with CAR T cell therapy. You can get a phenomenon called cytokine release syndrome. This creates very high fevers, massive drops in blood pressure. It can be lethal. It also, if you're killing all the body's B cells, healthy and unhealthy, you wipe out the body's ability to produce antibodies, so you have to support the patient with antibodies. And it can cause neurological toxicities, including delirium and seizures. The press have gotten hold of CAR T-cell therapy because it's exciting, because it sounds really clever, because it's new. But there are issues with it that still need to be ironed out. Issues to do with cost, issues to do with scalability, issues to do with the various side effects. It's still far from being a perfect treatment. So to summarize what I have talked you through, as cancer cells multiply, the ones that survive are the ones that can avoid the immune system or suppress the immune system. They fight back. Immunotherapies, the idea behind them is to create a cancer-fighting immune response, to overcome the barriers, to generate cancer-fighting T cells where there were no cancer-fighting T cells, or to disrupt the way that the cancer cells are suppressing the white blood cells. In the past 20 years, we have gradually been using more and more immunotherapy. Some of them are pretty standard treatments. Treatments like rituximab, which targets a protein called CD20, that's been approved for use since 1998. And there are many different licensed antibody treatments. The new generation of immunotherapies that are creating the current excitement work in different ways. I haven't talked about them today, but there's a whole group of immunotherapies called the checkpoint inhibitors. They are there to prevent cancer cells from suppressing white blood cells. Or we have immunotherapies like the CAR T cells, where you are deliberately generating a cancer-fighting army of T cells. As I say, these CAR T cell therapies, they're exciting, but they still have limitations. I'm going to stop there because I know time is tight, but I hope that has given you a useful introduction to some of the science behind cancer immunotherapy.